Hi everyone, and welcome to the chapters 8, 9, and 10 lecture from Crowley's An Introduction to Human Disease. All three of these chapters are about pathogens and communicable diseases. They go into a bit more detail than I think is necessary for this course, so I've tried to distill it down in this lecture to the key points that you should know. So, communicable diseases are infectious. Well, that's not true. They are diseases that can be passed from person to person. And most infectious diseases, as in diseases that are caused by an infection with a virus, bacteria, fungus, protist, helminth, or arthropod, most of them are communicable, as in they can be spread from person to person. There are some infections, though, that are not spread from person to person. One classic example is tetanus. So most infectious diseases are communicable. Usually we use them kind of somewhat interchangeably. So all communicable diseases, though, are caused by infectious agents, pathogens. And pathogens can be divided into different biological categories. We've got viruses, bacteria, fungi, protists, helminths, and arthropods. And we'll talk about all of these. But first, we're going to talk a little bit about the epidemiology or spread of communicable diseases. And so I'm doing chapter 10 first. So diseases, when they spread from person to person, they can fall under one of these three different sort of patterns. Okay, so you can have an epidemic, which is an outbreak in a localized area of a communicable disease, and the methods for controlling it are different if it's an epidemic, a pandemic, or an endemic, right? So epidemic is when it sort of kind of first appears, or when it appears after being gone for a long time. It's sort of an abnormal number of cases in an area. A pandemic is a disease that has spread globally. We're, of course, very familiar, having recently experienced a pandemic of SARS-CoV-2 or COVID-19. An endemic is when a communicable disease sort of becomes a normal disease in an area and is kind of the outbreaks are predictable. There's a constant spread. And the numbers are sort of constant and predictable, that it's just a disease that now is in that area. In this map, it shows dots in all continents, but a lot of diseases that are endemic to just certain regions, like sort of tropical regions. So like, for example, malaria is endemic in Africa and parts of the Middle East. All right, it's not in the US. All righty. So communicable diseases can spread a number of different ways. There are several modes of transmission. Some are direct, so they involve actual direct contact with the other person. Physical contact, so horizontal transfer is by physically coming into contact with another person, either touching them, shaking hands, or talking really close so that the droplets from their, mouth are actually coming into contact with you, and or sexual intercourse for sexually transmitted diseases in particular. Vertical transmission is when disease is passed from mother to child, usually through the placenta, through pregnancy, can also be through breastfeeding, though. But if it's generally, if it's mother to a child after they're born, we just call it horizontal transmission because it's a person touching another person. Indirect methods of disease transmission. This is when you never actually come into contact with the person who you technically got the disease from. These are when there's some sort of intermediary between. So fomites is a term for just inanimate objects that can hold viruses and bacteria and transmit them. They're touchable surfaces, I guess. So doorknobs is a classic. Fomite, towels, blankets, another classic one is one of an early historical account of bioterrorism when the American colonizers gave smallpox infected blankets, so blankets that had been used on smallpox patients, as gifts to the Native Americans on purpose, knowing that they were fomites, that they contained smallpox virus and would spread the disease, and committed a lot of genocide that way. Indirect contact communicable disease can be spread through food and water, contaminated food and water, by ingesting it, and you get sick that way. It can be airborne, and this is specifically for respiratory viruses that can spread in minute particulates in the air, and so they can spread in actually that's not true. It's all respiratory conditions. I was thinking it's not just viruses though. There's a famous case of Legionnaire's disease, which is caused by a bacteria, and the first outbreak of it was in a hotel in Philadelphia, and the air conditioning system had been contaminated, and so it was being spread throughout the hotel via the air conditioning. So people were just breathing it in. And then lastly, another indirect mechanism is through vectors. And in communicable disease, when we're talking about vectors, disease vectors, we're usually talking about biting insects. So like mosquitoes, ticks, fleas, sandflies. There's a number of them, but mosquitoes are by far the largest disease vector on planet Earth. In fact, fun fact for you, mosquitoes are the deadliest animal on planet Earth. Arthropods are animals. It's not sharks, it's not horses, it's not snakes, it is mosquitoes. They kill the most people. So we can think about, if we think about these different modes of transmission, that is also how we can think about how to prevent communicable illnesses. The way we prevent communicable illnesses is generally through trying to block modes of transmission. So we can block horizontal direct transmission by wearing personal protective equipment, PPE, so gloves, masks, condoms during sex. These are all physical barriers to protect us from communicable disease in a direct transfer. To prevent some vertical transfer, so mothers have to, of course, be very careful about what they consume or what they're exposed to while they are pregnant. But if they do have a disease that can be transferred horizontally or vertically, like HIV, for example, one of the solutions is to deliver the baby via cesarean rather than by vaginal labor, because some diseases can be prevented, or sometimes just getting the baby out before the disease can be vertically transmitted. To prevent fomite transmission, of course, sterilizing, sanitizing things, surfaces, blankets, and this can also be to help prevent horizontal transfer or direct transfer, sanitizing your hands before and after touching other people. Food and water, the key there is to wash and cook things well. Bacteria and viruses, most foodborne illnesses can be killed by simply heating to the right temperature, and so that's a classic way to prevent foodborne illness. To prevent air transmission, again, having good filters, air filters in like HVAC systems is key, but also you can do some personal protection by wearing masks to prevent release and also to reduce some of the breathing in of airborne particles. And then for vector transmission, there's some ability to control those vectors in the environment. So using pesticides, insecticides in the environment to try to control the amount of like mosquitoes, try not to leave standing water out, and try not to enhance conditions for my mosquito breeding, but also to personally protect yourself by wearing mosquito repellent or by wearing clothes or having mosquito nets. Various things to prevent mosquito bites will prevent mosquito-borne diseases. Those are all ways to help control sort of behaviors that we can do to help control and block transmission. But the most effective way to prevent any communicable disease by far is vaccination. So historically, vaccination has had amazing results at reducing incidence of diseases and has even allowed us to eradicate some diseases from the planet. So smallpox is no longer a disease in humans, actually in any animal. It is a human-specific disease, and through vaccination we were able to completely eradicate it from planet Earth. There are still samples of smallpox that are in like vials and freezers in a couple of high security labs in the world, but it has not been, we have not had a human patient with smallpox since 1977 or something. So what is vaccination? Vaccination is essentially, it's a training, it's like boot camp for your immune system. So a vaccine includes an antigen, which, remember, an antigen is something that generates antibodies, and viruses and bacteria and all of our pathogens are covered in antigens. All the molecules on them, a lot of them can serve as antigens, foreign substances that our immune system recognizes. So these antigens can be either in the form of the different types of vaccines we have. We have live attenuated vaccines, where these contain actual live viruses or bacteria, but they have been attenuated, which means they've been weakened. So they aren't able to establish disease. They are killed pretty quickly by your immune system. They're very weakened, but they are live and they can replicate a little bit, and they tend to generate sort of a very robust immune response because they are most similar to the actual pathogen. We can also use killed viruses or bacteria in our vaccines, so just their little corpses, our little microscopic corpses. That is safer than using live attenuated strains. The live attenuated strains can still cause disease in immunocompromised individuals. They can occasionally, very rarely, they can revert to a pathogenic strain. So while live attenuated works very well, it's also more risky. But some viruses and bacteria, some just don't trigger the immune system when they are killed. Killed vaccines are preferential. Even better are the subunit vaccines. I don't know why I wrote unit. Subunit vaccines. These are ones where it just contains a little piece, like a protein, a surface protein of a bacteria or a virus that can't cause disease because it's not even a whole pathogen. But it's enough to trigger your immune system to make some really good antibodies that will recognize that pathogen if your body ever comes into contact with it. And then lastly, the newest variety of vaccines are RNA vaccines, and RNA vaccines contain RNA, which goes into your cells, and then your cells produce that subunit, and they make it on their own. These are revolutionary in the sense that they're also a lot easier to make. RNA vaccines, they are harder to, they have much shorter shelf life though. RNA is notoriously fragile of a molecule. It's very unstable. It breaks down very easily, which is why The vaccines have a shelf life. The RNA vaccines have a shelf life. They have to be stored in the cold. And when I say they have a shelf life, I mean like a couple of weeks. And like once you open it, it has to be used that day, kind of thing. So they sort of hang around for a shorter time in your body as well. So another ingredient that I have here listed for vaccines is an adjuvant. So these little pink balls, you can think of those as adjuvants. Adjuvants are, you can kind of think of them as maybe like immune system irritants. So your immune system is not dumb. If you inject a killed virus into your arm by itself, your immune system is going to look at that virus and be like, well, that's not a threat. Like, we can ignore that. And for a vaccine to work, your immune system needs to go through the motions and be like, okay, this is a drill. Let's prepare. Let's make antibodies. And so an adjuvant is something that kind of irritates and stimulates the immune system so that it will respond to the antigens that are injected. Okay, and so a good vaccine, it stimulates all of the innate and adaptive arms of the immune system so that your body produces antibodies and memory B and T cells, so that when you do come into contact with that pathogen, you already have those antibodies and memory B and T cells, and you jump right into a secondary immune response instead of that long primary immune response with that lag phase. You jump into an immediate immune response and can neutralize that pathogen more quickly, sometimes such that you don't even get sick. Other times that you do get sick, but not as sick because you have a much more effective, your immune response is faster. Something to mention here, the difference between sterilizing immunity versus protective immunity. So vaccines, some do give you sterilizing immunity in the sense that, like, for example, the smallpox vaccine was pretty good at sterilizing immunity, meaning once you had been vaccinated, you did not get smallpox. It's very rare. There are some vaccines that work very, very well at completely preventing infection. Some, though, don't do a great job of preventing infection, but they do a good job of protecting from severe disease. We call those protective. So the smallpox vaccine was pretty good at sterilizing immunity. If you were vaccinated, you were not going to get infected. But the flu vaccine and the COVID vaccine are much better at protective immunity. So it's still fairly possible to get infected, but the seriousness of the disease is much lower, is definitely reduced. So vaccination and immunity in general is important because it helps to protect individuals, but it also helps to protect communities and populations. So if we have a community where everyone is naive, nobody has any immunity, which is what happened when COVID-19, SARS-CoV-2 first emerged. It was a brand new virus, so no one had immunity. And so it spread very rapidly because one or two people had it. All of their contacts were susceptible and ended up getting sick. Then, as people started to develop some natural immunity, some small portions of the population had COVID and recovered fine, but still there were large portions of the population that were not immune, and so they still were spreading the disease. Still, infected individuals could still spread the disease very rapidly amongst populations. When there is a lot of collective immunity, though, we call this herd immunity. So when something like 80 or 90% of the population is immune, if you have two sick people, if all of their contacts are immune then it doesn't spread to, it kind of stops the spread. And so these immune individuals, the blue individuals here in this population, 3 out of 4 of them stayed healthy because of these, all of the immune people around them kind of help buffering them from the few that are sick. So herd immunity is as important for protecting really a minority of individuals who can't become immune. But you have to have very large numbers, usually at least upwards of 80% of the population needs to be immune in order to provide that protection, that herd immunity. And it depends too on how infectious the infectious agent is. Does it spread to lots of people very quickly, or does it really require direct contact? So when it comes to controlling communicable diseases, there are several layers that are necessary. So each layer, wearing PPE, gloves and masks, cleaning and disinfecting and sanitizing, vaccination, all of these things are not perfect, right? So somebody who gets vaccinated maybe the vaccine doesn't work in them, or maybe the vaccine does work, but they still got exposed to a very, large exposure, dosage exposure, and still had breakthrough disease. You could wash your hands religiously and still get unlucky. So all of these things, all of these layers of protection are imperfect. But by stacking these layers, we greatly reduce the ability of the virus to break through, or the infectious disease to break through. So if you think of, it's called the Swiss cheese model. So if you're just vaccinated, but you're not practicing any other safety precautions, then you could still have breakthrough disease. If you're doing these other things and you're not vaccinated, you can still have breakthrough disease. So the important thing in order to minimize communicable disease is to do as many of the things, the protective things, as you can, because by stacking those pieces of Swiss cheese, the holes stop you stop having overlapping holes, and you end up with a solid wall. So I like that Swiss cheese model. So the more pieces of cheese, the less likely you still have any holes that go all the way through. Okay, so that's a bit about the spread of communicable diseases, how they're transmitted, and that's kind of referring to all of these different types of communicable diseases. Now we're going to talk more about the different pathogens. A little coffee break there. So a pathogen is anything that it literally means a disease agent, an agent of disease. Patho means disease, so pathology, the study of disease. So most pathogens that we think about are organisms that make healthy people sick. Like anyone can get sick. Those are our true pathogens. But there are also some organisms that typically don't make healthy people sick, but can cause disease in folks who are immunocompromised, who have maybe undergone chemotherapy for cancer, and that's wiped out a lot of their cancer cells, but also their white blood cells, leaving their immune system compromised. People with AIDS, with acquired immunodeficiency syndrome, are very prone to getting infections with these, what are called opportunistic pathogens. So these are pathogens that healthy people don't have to worry about, but people who are immunocompromised do. So we're going to go through each of these different types of organisms and some of the common diseases that they cause, starting with viruses. Now, viruses are the smallest. Viruses are technically not even living things. They're not, we call them microorganisms, but they're not organisms. They don't actually metabolize, have any genes for metabolism, and they can't replicate on their own. They can't respire on their own. They are just little, they're very small. They are very simplistic. So all viruses contain at their core some nucleic acid. Nucleic acid can be either DNA, they can have a DNA genome like cells do, like our cells do. They also have the ability to have an RNA genome. So they can have double-stranded RNA, they can have single-stranded RNA. So they're unique in the sense that their genomes can be made of either DNA or of RNA. The genome is encapsulated, is covered by a protein shell called the capsid. I mean, capsid kind of sounds like capsule, so that's how I remember it's this protein shell. And it's made of these repeating units, so viruses often have this sort of geometric shape, like a 20-faced icosahedral shape. They can have other geometries though too. And this virus over here also has a capsid surrounding its genome. Okay, now some viruses, that's it. They just have a capsid, and then they have spikes sticking off of that capsid, which are just additional proteins that help them to adhere or stick to a cell in order to infect it. And so we call these naked viruses. Some have an additional layer, a membrane, kind of like a cell membrane. In fact, it's taken from the membrane of a cell when it leaves, that has the spike proteins sticking out of that. So naked viruses and enveloped viruses, they have slightly different methods of leaving cells that results in their structure. So in naked viruses, they assemble inside the cytoplasm of the cell they infect, and they basically build up to a point that they bust the cell open, the cell bursts, and that's called lysis, and the virus particles are released without a membrane, so just naked. Some viruses leave the cell through a process called budding, where their spike proteins they send to the surface of the host cell membrane, and then instead of bursting the cell open, they sort of pinch off. This is called budding. And so when they pinch off the cell membrane, they end up coated with that cell membrane that's been decorated already with virus proteins. So the two different mechanisms of release, of leaving the cell, result in naked or enveloped viruses. So viruses, like I said, they are not living. They are not classified as living because they can't replicate independently. They completely hijack the cell. So I made this little cartoon here, all right, of like a hijacking situation. It's literally what viruses do. They enter the cell. First, they attach. So those spike proteins on the surface are important for recognizing specific receptors that they use to bind to or dock onto the cell, which then tricks the cell into swallowing it. And so the virus then enters the cell. The capsid protein falls away, and ooh, Amber Alert. There's an Amber Alert, y'all. Let's check it out. I don't know how to do that. I guess I'm supposed to go to my news. Anyway, we're just going to have to suspend our curiosity for now. I don't know, prison break, kidnapping. I don't think it's weather related. Alrighty. Where was I? Okay, the capsid falls away and the genome, the nucleic acid, the RNA or DNA is released into the cell, and the cell starts to read that RNA or DNA and follow those instructions and produce those proteins, those viral proteins. So the cell basically kind of stops doing some of its own stuff and just starts becoming a factory of virus production. So the virus doesn't have its own ribosomes, it doesn't have its own amino acids, it completely uses it's like it goes into a factory and says, stop making your product and start making mine. It doesn't have its own factory of itself. So the particles actually kind of self-assemble once the capsid proteins are made and the copies of the viral genome are made. They self-assemble into new virus particles, and then they are released either by lysis, like in this picture, or by budding off the membrane, which results in enveloped viruses. So some common viruses that infect humans that I will briefly tell you about. There are the herpes viruses. Now these are an interesting family of viruses because herpes viruses have an ability to, they can infect a sort of primary tissue like the skin, but then they can also infect nervous tissue and go dormant in your nerves. And so once you're infected with a herpes virus, you always have that herpes virus in your body. It can go latent. It has ability to hide out in a dormant or latent state in the nerves, and then it can reactivate. Sometimes it never reactivates, and some people it reactivates multiple times. So herpes simplex 1 and 2, these cause cold sores on the mouth, and also they cause genital sores as a sexually transmitted disease. And again, those can go away and come back. Epstein-Barr virus is a very common herpes virus. It usually just causes like a mild cold in kids, but it can, in some cases, particularly when it infects people in their late teens or early 20s, like young adults, they develop infectious mononucleosis, which is a more severe illness. And varicella zoster is the virus that causes both chickenpox and shingles. The fun story here is it used to be thought they were two different viruses. Varicella was the virus that caused chickenpox, and zoster was the name of the virus that caused shingles. And then they're very different presentations of disease. So chickenpox are like a bumpy rash all over your body that itches, and shingles is usually just along a single area, a single dermatome patch of skin. It can be anywhere on the body, and it doesn't itch. It's very painful. It feels like a burn. And so for a long time, doctors thought they were different diseases. And then once we had the ability to, the technology to study viruses, we realized, oh, they're the same virus. It's people who got chickenpox as a child, they still have that virus, and they just merged the names, hyphenated them to varicella zoster. They have that virus in their body forever, and it can reemerge as shingles. So chickenpox can be serious. It is sort of a systemic infection, so it causes respiratory illness and also the rash. It can leave permanent scarring, and in rare cases, the respiratory part of the ailment can lead to pneumonia that can be fatal, but it's very, in developed countries, it's very unlikely for it to be fatal in childhood. It's much more dangerous if people get varicella zoster for the first time as adults, because they have a very strong immune reaction to it, and they can suffer from that sort of cytokine storm, just a storm of cytokines being released in their body that basically gives them shock. Organs start to fail. So, and the shingles can be incredibly debilitating, and they can lead to permanent nerve damage that causes chronic pain, chronic nerve pain. And so a vaccine has been developed for varicella zoster. If you're an 80s baby like me, you probably had chickenpox as a kid, but if you were born in the 90s, you probably got the vaccine, which came out, I think, in 1996. So maybe 2000s babies were the ones that really got it. Human papillomavirus, HPV, is actually family of viruses, should be human papillomaviruses. There's like 60 of them, and most of them just cause skin warts. If you get a skin wart anywhere, hands, feet, elbow, whatever, that's likely caused by human papillomavirus. There are a handful of strains that cause genital warts, and also a handful of strains that infect the cervix and mucosal linings like anal mucosa, oral mucosa, vaginal mucosa that can cause cancer. We call them oncogenic. They can, just like we have oncogenes, genes that can lead to cancer, oncogenic viruses are viruses that can cause cancer. So the most common cancer-causing strains and the most common genital wart-causing strains, there's a vaccine for those. So the HPV vaccine actually is a vaccine for, I think now it's 9 different strains, maybe 6 different strains out of those 60 or so. So it doesn't prevent skin warts, but it will prevent the more serious forms of disease. Arboviruses. It stands for arthropod-borne viruses. It's a portmanteau. It's a mixture of words. So arboviruses are viruses that are transmitted largely by mosquitoes, and most of them are encephalitis viruses. So right now I think equine encephalitis virus, or triple E, is of concern in the Northeast as it is circulating. West Nile virus is another one. You can get them from mosquito bites, and they cause inflammation of the brain. Noroviruses, super common. If you've ever had a day where you were just throwing up constantly, might have been a norovirus. It caused the most common cause of stomach flu. And I give quotations because it's not a flu. Flu is influenza virus, and that's completely different disease, but we sometimes call this 24-hour stomach bug. We call it stomach flu, but it's not. It's the stomach norovirus. HIV, human immunodeficiency virus, is the virus that causes AIDS. So AIDS is acquired immunodeficiency syndrome, and HIV is a virus, and it's, I guess, kind of clever in the sense that the cells that it infects are our white blood cells, our immune cells. So it directly attacks our immune system, the system that is meant to protect us. And so it does so very quietly. Your immune cells, if your liver is infected, for example, then your liver will show signs of not functioning properly, and there's ways to sort of measure that. There's more symptoms, right? But there's not a lot of symptoms of having an infected immune system until you start getting more infections, until those cells start to die off in quantities that leave you immunocompromised. So HIV, a lot of times there's no symptoms until there are symptoms, which is why it's important to get tested regularly, because there are treatments. We actually have very good antivirals to keep HIV from progressing to AIDS. So AIDS is much less common now than it was in the 80s and 90s because of good drug regimens. And we do have some, there have been some experimental therapies doing like basically a full T-cell replacement, like a bone marrow transplant, that have cured people of HIV, but it's not really a scalable therapy yet. Hepatitis viruses. Hepatitis is inflammation of the liver, and there's several hepatitis viruses. There's Hep A, Hep B, Hep C, Hep D, and Hep E. A, B, C, D, E. Oops. A, B, C, D, E. The two that are the most concerning are these two, Hep B and Hep C. Hep B, we have a vaccine for, and it's part of the infant vaccine regimen. And Hep C, we do not, though in the last few years, we've actually developed an antiviral that's very effective at reducing the viral load, so helping to control that. But these two viruses cause like a majority of the cases of liver disease. And they also are oncogenic, like HPV. They can cause cancer of the liver. So those two are a very big concern. Influenza virus, this is the flu. and it is a respiratory virus, and it causes can cause serious lung damage and serious lung disease. Another important respiratory virus is respiratory syncytial virus. It's particularly serious in the very young and the very old. It's very dangerous for babies and for elderly folks, and is responsible for most of the hospitalizations of infants with respiratory illness. There is a vaccine now. It just came out. It is approved, not yet approved for infants. It is, oh no, it is. I take it back. It is approved for infants. And it's also approved for people over the age of 60 and for pregnant women. So pregnant women can get vaccinated and start making antibodies that get transferred through the placenta to the newborn to help protect them before they're able to get vaccinated. So they can't get vaccinated till they're like, I think 6 months or a year. Coronaviruses. Now, most coronaviruses cause common colds, but there are a couple that cause serious severe respiratory illness. So those are the SARS viruses. Severe acute respiratory syndrome is what SARS stands for. And there was SARS back in like 2009 in China. That was SARS-1. And SARS-2 is the recent pandemic. The COVID-19 pandemic was caused by coronavirus. SARS coronavirus 2. Poliovirus is nearly eradicated. It is not circulating anymore in most developed countries. I think there's only 2 countries in the world where it's still in circulation. We have done a great job with both with hygiene and with vaccination in reducing polio. But polio is a virus that it's actually it's foodborne, like you get it from drinking contaminated water, but it affects the nervous system, particularly neuromuscular system. So it causes partial paralysis of various muscles and not fun. It can also cause paralysis of the muscles that control your breathing, and that's how people die. Rabies virus is like one of my favorite viruses because it's like 100% deadly if it's not treated. It is zoonotic, so it's transferred from animals to humans through animal bites. So the virus actually, it's concentrated in the salivary glands. And so that when it gets transferred, the mode of transmission is through a bite, an animal bite. And then the virus itself finds its way to the nerves, and then it travels up the nervous system to the brain, where it infects specific parts of the brain, and it has specific symptoms like it makes you not want to drink water. It actually causes something called hydrophobia, where you're afraid of water, because water would flush those your saliva down and reduce the concentration of the virus. So the virus literally makes you not drink water to promote its survival. So the whole foaming at the mouth, that's classic of rabies. That's just because of extra saliva production, because that's where the virus is replicating, in the salivary glands. And it is 100% fatal through brain inflammation. And it also causes aggression to increase the biting, and even kind of like stimulates that bite reflex. So it's pretty crazy because it really manipulates the host to promote its survival. And it's 100% deadly without vaccination. So there is a rabies vaccine. It's not a fun one. It's usually given in the abdomen. It's 3 parts. And it's not a standard vaccine that everyone gets. You usually only get it if you've been exposed. So rabies, it is kind of a slow-moving virus in the sense that it does take time for that virus to get from the bite wound all the way to the brain. So it can take several weeks. And so there is time, but when you get bit, you can get vaccinated right then. The vaccine, takes 2 weeks-ish to take effect, to get your immune system prepared for that immune response to happen, and that is enough time to fight off the rabies virus before it gets to the brain. usually. So, and that means but you have to get that vaccine in a timely fashion. If you start having symptoms like the hydrophobia or the aggression, that means the virus is already in the brain. It's too late. You can get the vaccine, but if you still die. People still die. So it's like a nearly 100% fatal virus if you don't treat it right when you get bit. The rubella virus and the measles virus, these all do have are vaccine preventable. Probably no one in this watching this lecture right now has had measles or rubella. They're part of the MMR vaccine. Measles is a virus that causes a skin rash, but it's also respiratory and is particularly bad in kids. And before we had the vaccine, it was like the leading cause of, it can cause hearing impairment. And so a lot of times it caused deafness or partial deafness in infants who had measles. And it can also kill them through just respiratory issues. It's also a respiratory virus that happens to also produce a rash. And it's incredibly infectious. Like it spreads really easily. Very, very contagious. So that's why it's of such a big concern. Rubella is actually a super mild virus, doesn't make people very sick, but it's teratogenic. It causes birth defects. And so if a pregnant woman gets rubella, it can kill the baby or lead to numerous congenital defects because it affects cell division of the embryo. So, how do we treat viral diseases? Well, for most of them, all we can do is treat the symptoms. So, take over-the-counter medications basically to try to reduce your coughing or your congestion or your pain or your fever, and until the immune system actually gets rid of the virus. There's only a handful of antiviral medications that actually target the virus and prevent the virus from replicating. And it's very hard to design antivirals because viruses use our own cells. So it's hard to stop the virus when you don't want to poison your own cells, but that's really a lot of times the only way to sort of stop virus replication. So it's very hard to find a drug that is selectively toxic and only affects the virus and not toxic to our cells. So we do have some really good antivirals for HIV, human immunodeficiency virus. We have some good ones for herpes virus as well, for herpes viruses. So people who have herpes simplex might be taking these, or even if you have a bad case of, if you have shingles or chickenpox, they can give you it as well. The newest drugs are those for HCV, which is hepatitis C, and for, of course, SARS-CoV-2, which are not quite as good, I would say, as like the HIV medications, but they do reduce hospitalization. They reduce the fatality by something like 50%, I think. Don't quote me on that. So it's not 100%. They're not curative, but they do help. I want to clarify that antivirals are not antibiotics. Antibiotics are anti-living things. And so they're really, antibiotics are specifically what we use for treating bacterial infections. And viruses are not bacteria. So you cannot treat viral diseases with antibiotics. Sometimes people with viral illness will be prescribed antibiotics, but that's because virus infection oftentimes opens the door to bacterial infection. And so sometimes people have both. They might have both a virus and a bacterial infection. Or if they have a virus infection and they're very, very sick, doctors don't want them to get a bacterial infection on top of that. They might also give them antibiotics to prevent that. So you will sometimes get antibiotics when you have a viral infection, but it's not to treat the virus. I want to be clear about that. So because it's very difficult to design antivirals, because viruses use our cells, so it's very hard to target a virus. So because of that, we oftentimes we rely very heavily on vaccines, that we can protect ourselves from viral infection much more easily than we can treat viral infection. What's the saying? An ounce of prevention is worth a pound of treatment, I think. So vaccines are preventative. They prevent us, help to prevent us from getting sick or getting as sick. So you don't need treatment if you have, a lot of times it reduces the need for treatment if you have a vaccine. And we have a lot of vaccines for viruses. Here's a list of viruses that we have vaccines for. So that's a lot of diseases that we can prevent, and a lot of deaths. On to bacteria. Bacteria are cells. They are very tiny cells. They are very different than human cells. Humans are eukaryotes. We have a nucleus. We have different organelles. We talked about that in a previous chapter. Bacteria are prokaryotes. They do not have nucleus. They do not have organelles. So they have a very basic cell structure. They do have DNA as their genome. They have a chromosome, but it's not inside a nucleus. It's just freely floating around inside the cell. It's usually kind of bundled up a little bit to not be taking up the whole cytoplasm. And so we call the region where the DNA is bundled up is called the nucleoid, because it's nuclear, but it's not a nucleus. Okay, so the nucleoid is the bundled up chromosome. Bacteria also have additional DNA that's not in their chromosome, but that floats around in these just little circles, little extra bits of DNA that are called plasmids, that are, I kind of call it bonus genes. These are genes that they don't need for like metabolism, for everyday survival, for making parts of their cell. These can give them extra superpowers like antibiotic resistance, etc. And they can be easily transferred between bacteria, which is how things like antibiotic resistance can spread so quickly and evolve so quickly in bacteria. They have ribosomes. Of course, all organisms have to have ribosomes. That's how we make our proteins. They're involved for protein synthesis. And of course, everything is held inside the cell by a cell membrane or plasma membrane, which is a phospholipid bilayer just like ours, but slightly different, different flavors of phospholipids. Bacteria, since they are single-celled organisms, we are sort of protected. We have multiple layers as multicellular organisms. We have many layers of cells that help to sort of protect us from our environment, right? But bacteria don't, and so they have an addition. Most of them, like, I don't know, 90% of bacteria, have a second layer called the cell wall. And the cell wall is something that humans, we do not have. And so we'll see that when we talk about antibiotics, there's a whole class of antibiotics that target the cell wall because that is a specific feature of bacteria. There are also components of the cell wall that serve as PAMPs. They are pathogen-associated molecular patterns. They are unique to bacteria and not humans, so our immune systems, our innate immune systems, can recognize them. Some bacteria also have some extra surface structures. Not all bacteria have these, but they can have these pili, which are like little, they're not like hairs. They can be used for adhesion and sticking, but they also are used as almost like spikes. They can transfer DNA, specifically those plasmids, from one bacteria to another, so they can sort of like stick to each other, kind of dock on each other and transfer like a straw. It's like a hollow straw, and they can transfer materials between cells. A flagellum, some bacteria have flagella. They might have one, they might have several. And these are for locomotion, helps them move. Some bacteria don't have flagella at all and they can't move. They just depend on, you know, being carried by the wind or water or other organisms. So bacteria also have different shapes. Ooh, I gotta move my face here. They have different shapes. So we've got rod-shaped bacteria, which are called bacilli. We have spherical-shaped bacteria, which are called cocci. And most of the pathogens kind of fall into these two categories, but then there's also some extra special ones that have a spiral shape or a corkscrew shape. They can also have different arrangements. So cocci can be individual, they can be in pairs called diplococci, they can be in chains, they can be in clusters, and these are all just things that help to kind of distinguish them microscopically when we look at them under a microscope, because two cocci look very similar under a microscope. Streptococci means that they are cocci or spheres that grow in chains. Staphylococci means that they grow in clusters, and those are probably familiar names to you because of strep and staph infections. And we'll talk about some of the important bacterial infections coming up. So bacteria can have these different shapes. They also can have differences in their cell walls. So we said that most bacteria have a cell wall because they need that extra protection from their environment because they are just single cells, and having a single membrane between their insides and their outsides is just a little too fragile. So we can divide, most bacteria that have a cell wall can be divided into these two types, gram-negative and gram-positive. So they get the name gram-negative and gram-positive based on how, based on a staining technique called Gram staining that was invented or designed by someone whose last name was Gram. And so in the staining technique, the cells that are gram-negative end up staining pink, and the cells that are gram-positive end up staining blue. And if we look at the cell walls on a molecular level, the molecular differences between them are that gram-positives have a much thicker cell wall. And this cell wall is made up of a molecule called peptidoglycan. And it's the peptidoglycan that holds on to that purple stain, the blue dye. And so that's why they stain much darker, because they have a lot more peptidoglycan in their cell walls. They have a thick cell wall. Gram-negative cells have a thin cell wall, but they have an extra cell membrane outside of it. So they have two membranes and a thin cell wall, versus gram-positives have one membrane and a thick cell wall. It means that there are different susceptibilities of these different bacteria to antibiotics as well. Membranes are really good at blocking things from entering the cell. Gram-negatives tend to be naturally more resistant to antibiotics because they're just better at blocking those antibiotics from getting into the cell. Gram-positive cells are more sensitive in general to antibiotics typically, and they also are more sensitive to antibiotics that affect the peptidoglycan because they are so heavily dependent on their peptidoglycan layer. So gram-positive, gram-negative, just it's a difference in the type of cell wall that the bacteria have. Some other important features of bacteria: some bacteria have the ability to form endospores. So a healthy dividing bacterial cell, when it's under stress, like maybe there's, the environment is dry, or it's really hot, or there's not a lot of nutrients around, okay, it can go into a dormant, it can form this sort of hard shell around it and around its genetic material, and that is very resistant to various environmental conditions. And this spore it's no longer metabolizing, it's not making proteins, it's not respiring, it's just basically an enclosed genetic material that can reactivate when conditions are good, kind of like a seed. How you can have seeds for plants and you can store them for a long time, and then when you plant them, they germinate, right? So it's kind of like that. Some bacteria can do that, and it means they can persist in the environment for a long time and cause infection. Bacteria have different oxygen needs. A lot of bacteria are aerobic, like we are. They need oxygen, so they can only grow in high in environments where there's enough oxygen for them to breathe and respire. Some bacteria have evolved to be anaerobic. Their metabolism is completely dependent on not having oxygen around. In fact, oxygen is toxic to them, and so they cannot grow in environments where there's a lot of oxygen. They can only grow in a low oxygen environment. And then we also have microbes that are sort of flexible and can do both. So they're aerobic, but they also have the ability to turn on anaerobic metabolism and grow in low oxygen environments. So some common bacteria that infect humans that I'm going to try to go through somewhat quickly. Staphylococcus aureus, this causes our staph infections, which can be localized and superficial, like of the skin, but they can also progress deeper into tissues or infect the blood. We also have a lot of drug-resistant strains of Staph aureus, which means we don't have an ability to treat those infections, and they are much more dangerous. So staph infections are particularly dangerous because there's a lot of strains because they're common, and there's a lot of strains that we can't treat. So basically, in this age now of antibiotic resistance, we are kind of being reverted back to the days before antibiotics, where there are common infections that used to kill people before antibiotics existed are now starting to kill people again. Some other ones, Streptococcus causes strep throat. It can also cause other infections, but the most common one is strep throat. And also Streptococcus pneumoniae, which is a common cause of ear infections and of pneumonia. Neisseria gonorrhea, the disease is in the name, it causes gonorrhea, a sexually transmitted infection that can be asymptomatic, but it also can progress up the reproductive tract and cause infertility. Neisseria meningitidis is one of the causes of bacterial meningitis. The other one is actually Streptococcus pneumoniae. It can also cause meningitis. These are two. So we have a vaccine actually we have vaccines for both of them. But the meningitis vaccine is for Neisseria meningitidis. And there's a separate vaccine, a Streptococcus vaccine, strep pneumonia vaccine, that also helps to reduce meningitis, but also helps to reduce pneumonia. Some bacteria that are, some other ones here, diphtheria we have a vaccine for, it doesn't really exist so much anymore, it's very uncommon. Listeria is foodborne illness, so you get it from eating contaminated foods, but it can cause gastrointestinal disease, but it can also kind of infect lots of different cells in the body because it has this ability to kind of jettison itself out of cells and into other cells. So it can also spread across the placenta, and it can cause miscarriage. So it's particularly dangerous for pregnant women. Bacillus anthracis causes anthrax, which is a not terribly common illness, but is it can form spores. This is a spore former, and so these spores exist in a lot of soil, and the spores can also be engineered in a lab and then kind of used in bioterrorism, which is so it's a big bioterrorism concern. These Clostridium bacteria, all the Clostridium family bacteria, are anaerobic. They like low oxygen environments, so they tend to, so Clostridium perfringens and Clostridium tetani they infect wounds, basically, typically deeper wounds where there's not a lot of oxygen. And Clostridium botulinum is a foodborne illness that you get primarily from canned foods. They grow in cans where there's no oxygen. So if you have a canned food that the seal is popped or it's expanded, that's bad news. Throw that away. That can give you botulism. And Clostridium difficile is more popularly known as C. diff, and it causes infections in the intestines, and where there's not a lot of oxygen, and causes really bad diarrhea, and it can be difficult to treat and can be recurrent. It's actually, in a sense, it's a normal bacteria. Most people are colonized with C. diff, and it really becomes a problem after antibiotic treatment, when antibiotics wipe out all the other bacteria in the gut but don't affect the C. diff. The C. diff then overgrows, and that's bad news for the gut environment and leads to a lot of diarrhea, which can be dehydrating, can ultimately be deadly. Haemophilus influenza, not to be confused with the influenza virus. All right, this is more commonly referred to as HIB, H-I-B, for Haemophilus influenza B. We have a vaccine for it, so it's much more rare now. Yersinia pestis, that is the causative agent of the plague, the Black Plague of the 1500s. Actually, it happened a few times in history. It still exists. People do still get sick from it, but it is treatable with antibiotics, and we know much more about how it's spread, and we've improved hygiene and around fleas greatly, and so it's not nearly as big of a problem in the modern world. Salmonella, Shigella, Campylobacter, E. coli. These guys are all foodborne illnesses. They can make you sick, give you I don't want to say upset stomach. That's an understatement. But they can cause serious gastroenteritis, inflammation of the stomach and intestines. Some species, like of E. coli, for example, there's a lot of normal species of E. coli that don't make you sick, but there are a handful of pathogenic strains that have extra bonus genes. They've acquired plasmids that give them extra virulence factors, and they produce toxins that can make you very sick. So these are all important to watch out for. In foods, it used to be that the most dangerous foods were sort of uncooked meat that was contaminated with just a little bit of fecal matter from cows, because these are all actually normal intestinal gut microbes of cattle or birds, poultry. And so if in the slaughtering and processing practices, if any of those bacteria got onto the meat, then it could spread to the humans that consumed it. Now it's more of a problem in produce because of runoff from like cattle farms, big cattle farms. So there's all this cow manure that's got, bacteria in it, goes into the water, and then that water is used to irrigate the plants and water the plants. Then the plants end up coated in these bacteria. And so there's actually far more foodborne illness now in produce. So it's important to wash your produce because of that. Now, these guys, let's see, Treponema pallidum, this is the causative agent of syphilis. It's a spiral bacteria, and syphilis is a sexually transmitted infection. And then Borrelia burgdorferi causes Lyme disease. So these spiral organisms, one of the things that's kind of unique about them is they are bacteria that cause chronic infections. They go through stages. So there's an initial stage. There might be some initial symptoms. So for syphilis, there's actually like a painless canker, that like a blister that doesn't hurt though, that can form on the genitals. And sometimes people just ignore it because like, oh, what's that? It's weird. Maybe I should get it checked out. It's not really bothering me though. Oh, it went away. I guess I'm better now. And Borrelia burgdorferi, Lyme disease, the initial symptom is that bullseye rash, but not everybody develops it, or you may not notice it if you do. And it doesn't, sometimes it might be itchy, but it can also go unnoticed. And so there are initial signs of the disease, of the early stage of disease, when the organism first infects you. But beyond that, it can kind of go more systemic, so it can spread systemically through the blood and to other organs. And then at that point, it's very difficult to treat. So in these early stages, since it is a bacteria and we have a lot of antibiotics available, they are treatable. But once they progress to the later stages and spread more systemically, they are harder to treat, and sometimes not treatable, depending on the damage that they may have caused. So there's those guys. The Mycobacterium tuberculosis is the causative agent of tuberculosis, which Saranac Lake has a history of very tied to tuberculosis. It started out as a sanitarium, can never remember, curing, a place for people to cure, to come get fresh air into their lungs to help them cure from tuberculosis. And it's primarily a disease of the respiratory system, but you can actually get tuberculosis in other parts of the body. It's just more rare. So we can treat these bacterial illnesses with antibiotics a lot of times. And antibiotics are drugs that are selectively toxic to bacteria. And this is just an image from the text that shows you some different classes of antibiotics and where they act specifically in bacteria. So we've got ones that block cell wall synthesis. The original antibiotic, penicillin, targets the cell wall, and so there's a lot of classes of other antibiotics that also target cell wall synthesis, because cell walls are unique to bacteria. We don't have them. The enzymes in bacteria that do transcription and translation are different. So the ribosomes and the polymerases, they are slightly different. They have a different version than we do. And so we can design molecules that inhibit the bacterial forms of ribosomes and polymerases and not the human forms. And so we have those classes of drugs. And there's a couple of others that work as well. So when antibiotics were discovered, and penicillin was discovered in 1928 in the lab, but it wasn't available until the 1940s for human use, because it took a while to develop it. But starting in the 1940s, when antibiotics started becoming available to treat disease, they were considered miracle drugs. People survived infections that were a death sentence before, and got better in a couple of days. And so they were really considered these miracle drugs, and there was this, big rush to find more antibiotics, and we did. And but then in the 1990s, we really we kind of maxed out our discovery period of antibiotics. And we started, even before then, when we use antibiotics, bacteria, they replicate very quickly and they evolve very quickly. So every time they replicate, they have slight DNA mutations, and sometimes one of those mutations is lucky and results in resistance to antibiotics. And so now we are in this era. We were in an era of like the miracle of antibiotics, but now we're in an era of antibiotic resistance, where a lot of bacteria are becoming resistant to the drugs that we have, and they're not working anymore. So there's a big concern, and it's one of the top 10 global health concerns, is the rise of antibiotic resistant microbes. It's sending us back more than 100 years it's sending us back 100 years in time, to a time where we can no longer treat bacterial illnesses, which is scary. Other downsides of antibiotics: they can trigger allergic reactions when people are allergic to certain antibiotics. Dysbiosis is a term that means like an imbalance of bacteria in your body. So not all bacteria are bad. We have tons of bacteria living in and on our body that actually contribute to our health. When we take antibiotics, antibiotics selectively target bacteria, but they're not great at targeting specific pathogens. They just can target a whole class of bacteria, so that they might be good for gram-positive bacteria. They kill all the gram-positive bacteria in your body, which throws the populations of bacteria in your body, which can oftentimes, since you have most bacteria in your gut, in your large intestine, it can cause diarrhea. It can lead to C. diff, overgrowth of Clostridium difficile. It can lead to yeast infections, which we'll talk about coming up with the fungi. And those are all related to killing off sort of the good bacteria in your body with antibiotics, along with the bad pathogens. So how do we diagnose bacterial diseases? The sort of original and low-tech ways are to simply culture and do microscopy. So I still remember being a kid and getting a strep test at the doctor's office. I'd go in and they stick a Q-tip, sterile Q-tip in my throat, and then the nurse would streak it on an agar plate. And we'd get a call like 2 days later whether I had strep or not, because it takes at least 24 hours for the bacteria to grow. So it's a slow diagnosis. And so we try not to rely too much on culturing, but for some bacteria, that's the only way we can do it. And so sometimes it does take time to get a diagnosis with bacterial illness because you can't know until you can culture it and grow it. Sometimes you can do microscopy directly of the swab. There's, again, a lot of normal bacteria in the throat, so there would be a lot of other bacteria that would show up when you do microscopy, but sometimes there are body samples where that is appropriate to do microscopy right away on the sample, and they can do a gram stain, and they might be able to get a sort of preliminary diagnosis. So my daughter had an infection one time in her lymph node, and it became abscessed, and they had to drain it. And so they drained it, and they both set up a culture, but they also did microscopy of the drainage fluid. And they identified in the microscopy that it was a gram-positive coccus. But it could have been a streptococcus, it could have been a staphylococcus. They weren't sure, so we had to wait for the culture results to know for sure what it was. Then okay, so once you know what the bacteria is, just if you know it's staphylococcus, there are some strains of staphylococcus, of Staph aureus, that are antibiotic resistant. So oftentimes also in a clinical setting, they'll do what's called sensitivity testing, where they take that staph and they spread it on an agar plate, and then they put these little pieces of filter paper that have been soaked in different antibiotics onto the plate. And if the antibiotic is effective, it kills the bacteria and you get this clear zone around the disc. And so a nice big zone of inhibition, we call that, is a good sign. That means that the antibiotic works, that the bacteria is sensitive to that antibiotic. But if there's no zone of inhibition around it, it means the bacteria is resistant to that antibiotic. And so a scary looking plate is one where it's all bacteria and little white dots and no zones of inhibition. That would be a bacteria that's resistant to all the antibiotics. Lots of these clear zones is what you want to see. That means that it's sensitive to lots of different antibiotics. There are a handful of bacteria and viruses that we have developed rapid antigen tests, which work a lot like pregnancy tests, urine pregnancy tests. It's called a lateral flow assay, where basically you have lines, right? You read 2 lines means that it's positive and 1 line means that it's negative. And this is actually based on antibodies that are built into this stick that can recognize the antigens in the sample, and some little colored beads that localize to these spots, these strips, which make them, have a line or not. So these are great because they give you a quick answer, and they are really very sensitive and specific. But they are also sensitive to, like, if the pH is off in the solution or something, it can ruin the test. So they have to be done following strict instructions. And again, they're only available for certain pathogens like strep throat, strep pyogenes, for bacteria, COVID, or SARS-CoV-2 for viruses. So certainly not effective it doesn't work for everything. All right, so that's bacteria. We've done viruses, we've done bacteria. I know this is going to be a long one because I'm shoving 3 chapters into one lecture here. Feel free to pause and restart, pick up later. Fungi. Fungi are eukaryotic cells. They have a nucleus like our cells do. So fungal cells are much more similar to human cells than bacteria. Fungi, I'm not going to go through their cell biology, but they tend to grow in one of 2 forms. They can either grow as yeast, which are like spherical-ish cells that replicate by budding, or they can grow as hyphae, which are elongated cells. And it's these hyphae that give rise to the stereotypical mold. The fuzziness of molds is due to the stringiness of these hyphae, these elongated cells that grow in sort of a stringy fashion. Now, there's not a ton of fungal pathogens. Fungi we do actually have a lot of fungi that live on our body normally. They are called dermatophytes. Sometimes they overgrow and cause rashes or itch or irritation, but they don't cause like dangerous disease. So the 2 biggest ones of concern are Candida albicans or Candida albicans, which is the cause of yeast infections, or sometimes it's called thrush, and those can happen in the vagina, in the mouth, and even on the skin. And it's an overgrowth of this normal fungus that resides on our skin that leads to irritation, oftentimes results in itching. Ringworm is a misnomer. At some point in time, doctors thought it was caused by a worm, but ringworm is a fungus. It's actually a family of fungi, and they can infect different areas of the skin of the body. Some infect the feet, some infect the groin, the warm, moist, different. So there's different fungi and bacteria that live on your skin, because your skin has a bunch of different like microenvironments, kind of like the earth has like deserts and rainforests. There are deserts on your skin, like your arms maybe are like the deserts. They're exposed to a lot of sun, versus the rainforest would be like the moist armpits or something like that. So there's different species that live in different areas and can colonize them and over-colonize them, leading to some irritation or rash. Fungal infections, like true fungal infections that cause disease, are called mycoses. Myco just means fungus. So mycoses is just a term for fungal infections. It's a medical term. And the 3 biggest ones that are true pathogens are Aspergillus, which is a respiratory illness. You get it. And Aspergillus it's just found in the soil and stuff, and you can breathe in some fungal spores, and it can cause infection in the lungs. Histoplasma causes histoplasmosis, which is similar to Aspergillus. It's a soil fungus that can be breathed in. And then Coccidioides. This one is a fungus that grows particularly in certain regions, and in the US it's found in like the southwest, like desert areas, and it can be breathed in. The fever and the respiratory infection it causes sometimes called valley fever, because of the southwest valleys where it's found commonly. So all of these, you can breathe them in. They can cause respiratory illness, or sometimes they can actually cause inflammation of the nervous system of the brain, like a meningitis. but not super, super common and not super communicable. You don't tend to pass them from person to person. You get them from the environment, from environmental exposure. Parasitic diseases is, we refer, when we talk about parasites, there's sort of three different biological categories of organisms that we include as our parasites. Technically, viruses and bacteria are parasites because they feed off of us, but generally, sort of colloquially, when we think of parasitic diseases, we think of protists, helminths, which are parasitic worms, and arthropods, tiny little bugs, essentially. And parasites often have very complicated transmission cycles. So the parasites, a lot of times they're transmitted by a vector, some intermediate. Sometimes they're not. Sometimes they're picked up directly by a host, but sometimes there's multiple hosts. So like a tapeworm that you get from, that humans get from eating pigs. So the tapeworm has a portion of its life cycle that is in pigs and a portion of its life cycle that takes place in humans. And so a lot of them have multiple hosts. So just wanted to talk about the language. I wanted to have this slide to mention the language of vector. Vector is an insect that transmits it, some kind of biting insect, and hosts are organisms that the that the parasite undergoes like part of its life cycle and not just gets carried around. Okay, so protists, let's start with these guys. Protists are cool. They're really interesting looking single-celled organisms. So they are single-celled like bacteria, but they are not prokaryotic. So remember bacteria, they have very simple cells, no organelles, no nucleus. Protists do, they have lots of organelles. Sometimes they have more organelles than our cells do. And they are just very varied. So they look totally different from each other. They're just like little alien cell species and really weird, have really weird cell biology. Some of them have multiple nuclei. Like Giardia, I think, has 4 nuclei, not 1 nucleus, but 4. So they just have some really unique cellular characteristics, and they're all very different from each other. And they're very common in the environment, and most of them are pretty harmless. They just, feed small organisms in ponds, but some of them can cause disease in humans. So some that I'll highlight: Giardiasis, Giardia lamblia, which looks like this. This is Giardia in this picture as well. And it's a normal, actually, resident of intestines of a lot of wild animals. And so then it gets into water and like ponds and rivers and streams through rain. And if you drink water that is not purified, you can drink this Giardia and it will infect the intestines and cause chronic diarrhea and chronic intestinal inflammation. And it's not fun. Sometimes it's called beaver fever. Or traveler's diarrhea is oftentimes due to giardiasis. So just make sure you drink clean water to prevent infection with Giardia, and also Entamoeba histolytica, which is an amoeba that can be found in water. Which of these others? Fecal oral. Oh, Cryptosporidium also is another one that can be found in water. So those are ones that you want to avoid by drinking clean water. Malaria is one of the top 3 infectious disease killers. HIV, malaria, and tuberculosis actually kill the most people globally. Not in the US, but globally. And malaria is transmitted by mosquitoes, and it infects primarily red blood cells, and it's sort of a disease of the blood. It causes fever, but it can also cause like blood clots in the brain that lead to death. Some other ones. Toxoplasma. Toxoplasma is important for pregnant women to avoid because toxoplasma can be teratogenic. It can cause miscarriage or meningitis in the newborn. So you can get it from cats, actually. So cats are one of the hosts. Actually, cats are the natural host. Toxoplasma usually cycles between cats and rodents, is its normal cycle. And humans are actually just accidental hosts because we have domesticated cats and we have a lot of contact with cats, and the eggs, or not the eggs, but the toxoplasma is excreted in cat stool, cat feces, and so by handling cat feces, we can, as humans, end up infected ourselves. And then one of my favorites, I'm really into the super deadly and rare microbes, I guess. Rabies is my favorite virus, and my favorite protist is an amoeba called Naegleria fowleri, and it is an amoeba that can infect your brain through your nose. And this happens very rarely. There's only like 1 or 2 cases a year in the US. And it happens when people go swimming in fresh water, usually like lakes, in warm environments. So usually in the south, not so much in the northeast. So I'm not too worried yet. But with global warming, it'll probably become more common. And when you like jump in the water and the water rushes up your nose, all right, that pressure can actually push some of those amoeba somehow. It can allow them to cross the membrane, the mucous membrane, and then they somehow find their way to your brain and literally eat your brain. And it's 100% fatal. Super, super rare, because if you think about the millions of people that swim in these warm water environments and only 1 or 2 develop this disease per year, it's a crazy low occurrence rate, but a very high fatality rate. Lastly, we have the helminths. Actually, that's not lastly. Next we have helminths. Helminths is the word for parasitic worms. And there's lots of different types. There's different types of parasitic worms. Cestodes are the tapeworms. Nematodes are the roundworms. And trematodes are flukes or flatworms. And they cause actually a ton of disease globally. And I like this picture because it gives numbers. So Schistosoma mannsoni is actually what I did my graduate research on, and it infects about 200 million people a year. Echinococcus, which is a type of tapeworm, affects over 100 million people a year. Ascaris, over 1 billion people a year get Ascaris. So helminth infections are particularly a heavy burden in undeveloped countries. A lot of them come from the soil or from eating contaminated foods. And so they are oftentimes more endemic in underdeveloped countries. And those countries also lack the resources of treatment. There are anti-helminthic drugs. There are drugs you can take to cure from or to treat a worm infection, but there's no long-lasting immunity. There's no real protective immunity from worm infections, so you can just get reinfected. You can treat it, but then you ingest worm eggs and you just get reinfected and you have to get treated again. And so that's one of the problems, is a lack of access to medication, but then even if they are medically treated, they can just get reinfected. And so helminth diseases are often referred to as neglected tropical diseases because they are common in tropical regions, largely underdeveloped countries, which don't have the finances, the financial structure, or the infrastructure to deal with them the way that we have in developed countries. And so there's a very heavy burden of disease, but they're neglected by countries that have the money and the funds to research and treat them. So there's a sort of niche group of researchers that are really committed to researching and trying to solve the problem of these helminth the heavy burden of Helminth diseases in those countries. A lot of these cause intestinal illnesses. Some of them, schistosomes, infect the blood. Those are blood flukes, but like tapeworms in your gut. And a lot of these, like Ascaris, is also, so a lot of them are ingested. They are ingested either from eggs that get under your nails when you're working in the soil and then you eat food, or from the food itself from animals that are infected. I won't go through, they have very complicated life cycles, so I won't go through those or even any of these specific ones. I just kind of wanted you to know more broadly. And then lastly, the arthropods. These are like tiny little bugs, little insects that can infect either be ectoparasites. So lice are ectoparasites. Scabies, technically, these are ectoparasites as well. So these are not infecting our internal structures, they are infecting externally. So lice live in the hair, they lay their eggs, and they walk around and eat dead scalp cells. So they are itchy, and in order to clear the infection, you have to make sure you get rid of all of the eggs, or the eggs will hatch and then you still have lice. And the eggs are sort of kind of glued to the hair shaft, so you have to use a really fine-tooth comb to really make sure that you get all of those little eggs or nits out. And you can, the adult lice can be killed with like a chemical shampoo, but also through physical removal. And nits can also be shed onto like pillows and blankets, and so those have to be sanitized. Usually just a heat cycle in the dryer is enough to kill them. Lice can't live very long, not on a human, so another way to treat like blankets and pillows and stuffed animals is to simply put them in like a garbage bag and put them like on your porch or somewhere, just not in, or just like in a closet, and just leave them there for like a week and any lice will be dead. Scabies is a skin infection, so these are little tiny mites that can burrow into the epidermis and they cause inflammation. They just like live in these little burrows under your skin, and it can leave these kind of track marks and areas of inflammation where your immune system basically reacts to like chemicals they're secreting that make your immune system angry and lead to this itchy rash on your skin. So we can treat parasitic diseases. It is harder because, again, these are eukaryotic organisms, so a lot of their biology is similar to our biology in the sense that chemicals that are toxic to them can also be toxic to us. So anti-helminthics, antiparasitics, they tend to have more toxic side effects than something like antibiotics. But it's sort of a short-term toxicity to kill that parasite, and it's generally worthwhile. Sometimes for parasitic disease, we'll treat them simply by physically removing them, like in the case of lice, or even some worms might actually be physically removed. There are no vaccines, though, for any parasitic illnesses, and there are people trying to develop them, but again, we don't also have very good like permanent immunity to protists and helminths. We can get reinfected and reinfected. There's not a good memory immune response that prevents them from colonizing, and so it's probably unlikely that we will ever have vaccines. That will probably not be a method we can use to control these larger organisms like we can with bacteria and viruses. I didn't mention vaccines for bacteria, so I am going to go back real quick because I did talk about them a lot with viruses, but I just want to point out we also have vaccines for a lot of bacteria. I guess I did. I mentioned that we have vaccines for meningitis, for diphtheria, for Hib, Haemophilus influenzae. We have a tetanus vaccine. There is actually an anthrax vaccine, but it's not given widely. It's just usually given to like military personnel. And There is also a tuberculosis vaccine. So vaccines do help to reduce a lot of the burden of these diseases. They are the best method for controlling infectious disease, but they are not always easy to design. Sometimes we get lucky and the first try we get a vaccine that works, that we pick an antigen that works really well, but sometimes it's easier said than done. And a classic example of that is for HIV. We've been working on a vaccine for HIV for decades, and we still don't have one. And there have been multiple vaccines that were developed and tested, and some of them actually increased infection. So that one has been a particular struggle to get a vaccine that actually works, and partly probably because HIV infects the immune system. It makes it harder to design a vaccine against it. But that is a long lecture, but it was 3 chapters, so I think in that case it was appropriate. Thanks for watching.